This is Dr. Double B, and this is my lecture on molecular pathology of CNS tumors, neuropathology. So here's a little table. I think this one's borrowed from WHO, showing some of the basic changes just as a reference if you want it, or you can find these online or in different study guides and things. So here's an outline of the actual content. So there's obviously a lot of things, as you can see from the last few tables. So we'll just cover some of the basics like IDH mutations, oligos, some of the H3K27 stuff, MGMT promoter methylation, and then methylation profiling, which of course I think is the most interesting. So this was just a random example of a neuropath sign out that I'd seen, neurocase sign out of a lot of the IHC and other stuff, and I just pointed out here because I think it's a pretty good description of some of these things. So like using basic terms like positive, negative can be a little bit confusing. So here they're saying negative for mutant protein, all those are mutant specific antibodies. Here intact nuclear expression for all the MMR proteins. So there's kind of some good ways to describe these antibodies that are a little more molecular focused or IHC antibodies. Alright, so let's talk about astrocytomas. So of course the biggest thing to talk about here is IDH1 and 2, isocitrate dehydrogenase. So these are seen in the adult type diffuse gliomas, astrocytoma, IDH mutants. They're all named after the mutation. Those can be various different grades, 2, 3, or 4. Then there's oligodendrogliomas, which has all the molecular changes in the name, IDH mutant, and 1p19q codeleted, and that can be grade 2 or 3. And then glioblastoma, IDH wild type, which is always grade 4, high grade. So isocitrate dehydrogenase is part of the Krebs cycle over here. Well, technically IDH1 and 2 that we talk about, which are in the cytosol and mitochondrial, are not really part of the Krebs cycle. They do the same conversion that's done there, but they don't technically do the Krebs cycle part of it for energy production. IDH3 is the one that localizes to the mitochondria, so it's not on the mitochondrial genome either, but that one's part of the Krebs cycle. So just a little technicality. So here's the chemical conversion that happens and that it catalyzes. So this one you can find, and I kind of drew it out. I was thinking about, animating these, and the IDH1 mutant catalyzes a different conversion. So the main one is, or the normal one is isocitrate to alpha-ketoglutarate. Again, the same thing that happens in the Krebs cycle. And then the mutant, couldn't find the exact chemical conversion, but basically it converts that to D2-hydroxyglutarate. Yeah, I was thinking about animating it, but I realized, no one really cares about the chemical conversion. It's basically just magic, so we might as well just make it magic.
My wand! Look at my wand! Ferroverto.
That wand needs replacing, Mr. Weasley.
Alright, so run is our IDH1 here. Hopefully this video doesn't get taken down because of this. It's fair use, right? Alright, so here we have our normal wild-type IDH1 converting the isocitrate into alpha-ketoglutarate. Now we have the mutated IDH1 with the broken wand converting the isocitrate into D2-hydroxyglutarate. And so this is going to accumulate within the cells. So again, for reference, here are those two things. So there's a normal IDH1 isocitrate to alpha-ketoglutarate, and the mutated IDH1 converting it to D2-hydroxyglutarate. And a good chemist might be able to tell you that's going to interfere with the alpha-ketoglutarate and affect cell growth and act as basically an oncometabolite. So a little different than the way some of the other oncogene tumor suppressor things work classically. So we used Cosmic for some of these other things as we've gone through these. So you can see most of these are one mutation again, this R132H, like you see with a lot of kind of oncogenes, although it's a little different in this case. There are some rare ones that you can see up here in a smaller percentage. And then another small subset is IDH2 mutations. And so this can be detected molecularly by sequencing assays like NGS or more focused ones, but it can also be detected by IHC, and this is a mutation-specific antibody. So you're not just looking for IDH1, because that's always going to be expressed. You're looking for the specific mutation. So there's an antibody for the R132H. So it's only going to be positive if it has that mutation. Except, sometimes there's some cross-reactivity, so the R132L could also be positive as well. And like a lot of things, a lot of mutations that we look at in molecular, there are targeted therapies for these.
At least no one on the Gryffindor team had to buy their way in. They got in on pure talent.
No one asked your opinion, you filthy little mudblood. She'll pay for that one, Malfoy. Eat slugs!
You okay, Ron? Say something.
Okay, that's enough of that. So yeah, targeted therapy, vorasidenib or voranigo, if I'm saying those right, can be used to treat grade 2 astrocytoma or oligo with a susceptible IDH1 or 2 mutation. And again, small molecule inhibitor, like a lot of these. So another big change you see in the same category, the IDH mutant astrocytomas, is TP53 mutation and loss. And I've talked about this a little bit in another lecture, the first one, but often referred to as guardian of the genome, activates DNA repair proteins, arrests cell growth, initiates apoptosis, and there's an animation elsewhere on YouTube and in the first lecture if you want to see that in more detail. And in a lot of tumors you'll see TP53 loss, and including these IDH mutant astrocytomas. And these can be a little tricky. These are, again, the same examples of ovarian serous carcinomas where you can have the wild-type low-level expression, you can have complete loss, and then you can have diffuse expression of a mutant protein. And it can be a little tricky. This middle one, the complete loss, I think is extra tricky in brain because it's a little hard to tell what every single cell is, if it's a tumor or not. So it can be difficult to distinguish wild-type and complete loss. So another big thing you see in the same category is ATRX loss. So transcriptional regulator, part of the SNF family. It's named after the syndrome, and there's an IHC for it again, so you can see loss in the tumor cells. And then here we have some background normal, which do look like not atypical cells compared to larger atypical tumor cells here. So again, a little bit of a summary of that. If you're, and specifically in grade 4 astrocytomas, your choices are the IDH mutant, grade 4, with a lot of these changes we talked about, and then there's the IDH wild type 1, which is worse prognosis. The mutant one was formerly known as secondary GBMs, and the wild type ones were primary, but it's not really used. And when you're in this category of IDH mutants, one of the molecular changes that can upgrade it to grade 4 is if you have CDKN2A or B homozygous deletion. And this is an example of fish, and I forgot to show where I got some of these images, but this would be normal, where you have 2 copies of the red, the CDKN2A, and then the green, which would probably be a control, or I guess it could also be CDKN2B. And then here you have the red missing, so it's a hemizygous deletion, just one, and then what you really want to see is the homozygous deletion. So fish has some advantages over like NGS for this, where it can be hard to tell if it's hemizygous or homozygous. In the fish you can actually look at individual cells and see if it's 1 copy or 2. So a little bit more, here's a little bit too much information at once, but here's some details on the IDH wild type glioblastoma. So if you do the IHC for the IDH1 R132H, that's considered adequate to rule out that it's an IDH mutant in patients over 55. If they're less than 55, or if they have ATRX loss, which is suspicious for the IDH mutants, then you got to do the sequencing to make sure you're not missing anything. These will still, again, like a lot of tumors, will have TP53 mutation loss, so that's not really specific for mutant or wild type. It'll also often have the CDKN2A or B loss, or some other alteration in that same pathway. And then here are three of the important ones. So these can actually upgrade to IDH wild-type glioblastoma if you're thinking about other things. If you have TERT promoter mutations, EGFR gene amplification, or gain of 7, loss of chromosome 10. So we'll talk about methylation profiling at the end, but that could be used in diagnosis, and it has some molecular subgroups. And BRF-V600E can be seen in epithelial GBM, but there are some catches that, maybe some of these are getting reclassified. Maybe that's anaplastic PXA, which I'll also show a little bit at the end. There are also some EGFR variants. Most common is this EGFR V3, where you have this deletion of exons 2 through 7 at the right here. So you kind of have this loss of a big region here. So it doesn't bind the normal ligand, but it does have, again, low-level constitutive signaling, like a lot of our oncogene alterations. So you have amplification, and you can have this as well. So here's some histology examples. So here's an IDH mutant. This is grade 4. Some atypia, you might have to look around to kind of confirm the grading. So here's the IDH1 R132H IHC. So it's positive staining, so you could say it's positive for the mutant protein in this case. P53, here we have that diffuse wild type, so it's most likely a missense mutation. And then some probably normal cells in the background. And that can also kind of give you an idea of your tumor cellularity. And then ATRX, where you have the loss. So the negative cells are the ones with the loss here. And then the staining cells are probably, background normal. So here was an IDH wild-type glioblastoma. Histology here, negative for the IDH1 R132H. Again, you might have to do sequencing to prove it's not one of the other ones, depending on the age and other things. ATRX is intact here. And this is where you can see it's a little bit hard to tell, is it some background, is it wild type? You have some staining here, so maybe it's wild type, but then we know we have some normal cells in the background probably, so it could be those two. So it can be a little tricky to tell apart wild type versus complete loss. Alright, so next tumor category is oligodendroglioma, with all the molecular changes in the name pretty much, so fried egg appearance here. And so one of the ways it can be detected is FISH fluorescence in situ hybridization, which are probes to specific regions. Of course, it's more than 4 base pairs, it'll be thousands of base pairs. But then you can look for presence of things, deletions, also, well, not in this case, but in other cases, things like translocations. So here's a little cartoon of it. So you could technically maybe put, 1P in one color and 19Q in the other color and look for that codeletion, but we like to have controls, and it actually matters. Classically, you should just see that arm. So it's nice to see that the other one is still intact. So here we have one FISH reaction would be with probes for 1P and 1Q. And so you want to see that 1P deletion. And then you also want to see the 19Q, which was part of the same animation, so that disappears as well. So this would be a positive case for 1P 19Q codeletion. And then here is just an actual image, basically the same thing, where you can see the probes up here, 1P, you see loss of one of the reds, and then 19Q in green, so here we see loss of one of the greens, loss of a green, loss of a green. So FISH is probably the most common, but maybe not the best. So this was pulled from a paper, again I forgot to give the reference, but you can probably find it, where just kind of shows one of the pitfalls. So this is a case of oligo, so everything's good in this one. So you haven't seen these sorts of arrays, it's basically showing you copy number where this would be normal. So we have normal 1Q, normal 19P, and when you see a drop, it's a loss, and so here would be one copy loss of 1P. And then same thing of 19Q. And so this is what you're supposed to see in the oligos. You're supposed to see complete loss of that arm, and then the other ones are intact. And so the little bars here are showing where the FISH probes are. So they're to a specific region, and usually it works fine. You'll see loss of the probe if you have loss of this whole chromosome. So here we have loss of that, loss of 1P, and loss of 19Q. Now, here is again, this is showing a pitfall case. So we have a glioblastoma, IDH wild type, and here we have the normal level. There's a little variation here, maybe some other low-level alterations. But we have some changes in 1p19q, so we have this very focal deletion on 1p that happens to be in the region of the probe. And then we have another focal deletion that happens to be in the region of the probe. So this would basically be a false positive, where it would get called as 1p19q loss, but it's not really an oligo. So hopefully they would notice something is off and correlate that with histology and other findings. But here's where maybe the array is a little bit better. Again, just showing loss of that region on 1p and loss of that focal region on 19q. And here was an example of that. So again, at the top left, some histology, a little bit of the Friedegg appearance. So it'll also have the IDH1 mutation. This would be maybe some wild type staining for p53 here, and then intact ATRX. And then the 1p19q you would see by FISH or other methods. Alright, so next category is these H3 K27 altered tumors. So this is part of histone regulation. So the SNF complex, we've seen other things also, is involved in chromatin remodeling, repositioning nucleosomes, and basically making DNA accessible for transcription factors, kind of unwinding it from histones is how you think of it, and I have a little video of how that happens.
Keep my DNA out your f***ing nucleosome.
I'm going to, okay?
Alright, so that's how it happens. Alright, so here's another slide with a little bit too much information, but kind of a lot of the facts that go into it. So again, we have the H3, which is a thing that kind of ends up getting altered. So that's the cursor rock here, and then the trimethylation actually protects it from the SNF or the Will-Smith complex, so that it can't get to it and can't kind of activate transcription. So this trimethylation is done by the EZH2. You can see that here, and this is called the PRC2 complex. This is the whole thing, it's a polycomb complex. So these different proteins. H3 is a histone, has some different variants. Now you might see one tricky thing. Sometimes you'll see this as K27, sometimes K28. When we do the stains and things, it usually says 27. So the thing is, the initial methionine will be cleaved on this and other proteins. This happens as well. So different people describing this use different sites. So if you were to see a molecular sequencing result, this would be 28, the 28th amino acid. But when we talk about the IHC and things, they use 27 because it's the 27th once that methionine gets cleaved. But basically it refers to the same thing. So you can use kind of whatever you're using, whatever it's called. And so this ME3 refers again to the trimethylation of K27, which affects this regulation, prevents that SNF-Will-Smith interaction. And you have loss of this in diffuse midline glioma, which is caused by most commonly a specific mutation. And it gets a little confusing because you have this M, this ME3. So this refers to the specific mutation, K to M. It's amino acid 27 on the protein, or 28 if you're counting on the genome. Rarely another one, K27I or K28I, and then rarely overexpression of this EZHIP, which is a competitive inhibitor of that PRC2 complex, so prevents it from working. So hopefully that makes sense. That one's a lot of information. So here's an example of that. So again, histology up here, it's not going to have the IDH1 mutations. And here's where these two stains, you kind of got to keep them straight even though they look a little bit similar. So this is an antibody to this specific mutation, this K27M or K28M. And it's positive nuclear staining here, which means it does have that mutation. So we would say positive for mutant protein or something like that. And then here, the side effect of this mutation is you have loss of this trimethylation. So here we're actually looking for loss of staining. So these positive ones, staining ones, would be the normal cells. And then these would be the tumor cells that have the loss. So we'd say there's loss of H3K27Me3 staining. Alright, so hopefully we're good at this point. We're going to move on to MGMT promoter methylation, and I have a lot of the same slides in the techniques lecture. I'll include it here for completeness for anyone who's just watching this one. But MGMT is one of the big things we test for in brain tumors, astrocytomas. So it stands for 06-methylguanine DNA methyltransferase. So it's a DNA repair protein that repairs alkylating damage to DNA, but the problem is it'll also repair the effects of alkylating chemo. So I use Peter Griffin, thinking of it as like a dumb tumor suppressor, although I guess it's just doing its job. So the promoter for this can become methylated in a lot of these tumors. So it happens at the CG islands, and then will shut down transcription of the downstream MGMT gene. And here's just a random image that shows some of these different methylation things. So there are different genes, proteins involved, and different things that can happen. That was just to make a fellow happy. Alright, so here's MGMT doing its job. Here's alkylating damaged DNA by the little green squiggle there. The MGMT will repair it, which again is normally it's doing its job. So if there's a tumor and we're giving an alkylating agent, if MGMT is functioning, it'll repair that, and so the chemo won't have its full effect. Things like temozolomide. So if the promoter is methylated and it's not expressed, that's a good thing because it's going to allow the alkylating agent, the temozolomide, to have its full effect and kill off the tumor cells. It also gives it some kind of differential effect, so the tumor cells that don't have the MGMT will hopefully be the ones to die off, and the normal cells that have it will be able to repair it better. So the classic way to detect this is pyrosequencing. So it's used for cancer hotspot mutations, and also, again, for promoter methylation testing, for like this, and MLH1 promoter methylation. So here we have sequence, and I'm showing two slight variations. Again, in most cancer sequencing, you'll usually have a mix of some tumor and normal. This is just kind of a random sequence here. So we add one nucleotide at a time. So in this case, we add a G first, and when a nucleotide gets incorporated, a pyrophosphate gets released, and that's another normal thing that happens. But in the mix, we include the sulfurylase and luciferase that convert the pyrophosphate into a photon, a flash of light. So when something gets incorporated, you have that pyrophosphate release and that flash of light. And we monitor it. We have a dispensation order, so we know which order we're adding the nucleotides, and this kind of has to be carefully planned to cover whatever mutation or other alteration like methylation you're looking for. And so we see the peaks at the bottom when we detected that light flash. So we had a C, it doesn't bind, so we don't see a flash here. Now we had a G, it does get incorporated, so again we see a flash there. Now we have three T's in a row, so we're going to see all of these get incorporated at once, a lot of pyrophosphate released, and a lot of photons released. Then that one doesn't get incorporated, so it's flat line again. The T does, so again we see our flash of light. Again, here we have an alteration, so some of them have a C, some have a T, so the G gets incorporated to half in this case. So we see a peak that's half as strong because you're getting half the photons released. And then we had an A that covers the T, and again you see that half-size peak, and that's again where this has to be planned to cover any known alteration. So then we have a normal size one, and then I have just another kind of funny alteration at the end, where I'll have a unusual amount of G incorporated, and then the A. So it can be a little hard to interpret for certain things. And then here's another example, again part of the techniques, just if you haven't seen this before. So you would read off the peaks as the sequence. So this would be normal BRAF in the region of the V600E, but this is the wild type, so this would be GTG, AAA, there's a triple height peak here, TCT. so this is a V600E where you have a T to A mutation, so we're also getting an A here, and this peak is actually dropped because a lot of the strands are A now instead of the T. So these would kind of together add up to 1, or 100% of a nucleotide. Alright, so now on to the methylation part. So again, we're showing two different things here. So one where we have methylation of these two Cs, and then the other strand where we have none. So we treat with bisulfite, which converts unmethylated Cs into Us, or uracil. The methylated ones get protected. So in the unmethylated strands, we're going to have all of the Cs get converted into Us. Then we're going to do a couple rounds of strand synthesis. The U's become A's, the A's become T's. So then the final analysis, we actually do pyrosequencing and then look at whether these bases are C's or T's. If they are C's, then that means they were originally methylated and they were protected from that bisulfite conversion. If they're now T's, then that means they were not protected, they got converted to U, and then A and T, just to make it a little bit confusing here. And so then again, we do the pyrosequencing and look at whether we have a C or a T. C means methylated, T means unmethylated. And as always, we're usually going to have a mix of both things. Even when it's positive for the methylation, you're still going to have some unmethylated. And so this would be a negative case. So again, all of these are marked in this light blue here, the sites of interest. So these are CG islands. So you're going to see G on the other side of these. And then we're looking at whether it's C or T. So it was originally a C, part of a CG island. We have added the bisulfite, and this C got converted to this T. So now it's all T, so it's unmethylated. This C also got converted to this T, so it's unmethylated. This C got converted to one of these T's up here. If you look down at the dispensation order, you can see there are actually 3 T's right before it. The Y means it's one of the possible methylation sites. It's either a C or a T now. But again, we already had 3 T's, so it would have been like this high, and now that got converted, so now it's 4. And then this is kind of a normal one where it's all T. This is another one where, if you look down here, there were 2 T's already, and when it got converted, now we have 3. And then this final C here is kind of an internal control. It's not part of a CG island. It's not a possible methylation site, so it should always get converted to this T here. So that would be, if there was a peak here, something's wrong. It would mean the bisulfite had expired and wasn't working or something. And so here's a positive case that does have methylation. So we're seeing a mix of both. So we're seeing some that's unmethylated, but we're seeing these clear peaks that are methylated here. So we have some C on the first one, the second one, third one, fourth one, fifth one. And this worked, so this got converted. Again, it shouldn't be methylated, so this is normal. And then you can do an area under the curve, get an average, and report that out. But basically it would be positive. Alright, so that's kind of the older methylation, just looking at specific genes like MGMT, MLH1. But now the new, more kind of exciting thing is methylation profiling, looking at methylation across the whole genome and then trying to classify things from there. So if you think about it, when something becomes a tumor, you're probably going to have different methylation than normal. Some of it might be things that are obvious, like maybe you're going to have more tumor suppressors being methylated, not expressed. Maybe you'll have more oncogenes being unmethylated and expressed more, kind of driving the tumor. And then, of course, you're also going to have cell type specific expression. So different things are going to have different expression levels. I think this might have been a gist. We've got to label these. So you might have expression of KIT if this was, a chordoma. So you're going to have different expression of different genes based on the cell type of origin and the tumor type. So the idea here is we test a large number of sites, which is 400,000 or 800,000 as these methylation arrays have grown, and then throw some AI at the results, and then classify the tumors, if everything works, obviously. So these standard arrays work in a similar way. They still get treated with bisulfite first, and then you just figure out if all the sites are now a C or a T. and that's done by a microarray. And so I have a little graphic for microarray. This one isn't specifically for methylation, so these are going to be different nucleotides, but just use your imagination and pretend they're all looking at C's versus T's. So the general SNP array for single nucleotide polymorphism it'll be specific to a certain sequence. So you have all of these little oligonucleotide dots over a slide. It'll be a certain region, but then you'll also want to know, is this end, an A or a T? So the blue ones are supposed to be the same up to this point, and then we want to know, is it A or T? So we'll add label probes, or no, sorry, William. The things that are attached are basically the labeled probes, or the probes. Then the patient DNA is labeled and then binds to whatever it's complementary to. So in this case, as the T, it binds the A, and then you'll see this dot fluorescing because the patient DNA is labeled. And so you know that they had that SNP. And then this was showing a heterozygous example where we're looking for, we have here the G or the A, and they have some DNA with both, so it binds to both, and we see a weaker signal from both of them because there's not quite as much binding. And again, for the methylation arrays, basically same idea, but you're looking at all these sites to see if it's a C or a T after that bisulfite conversion. And so the results look something like this. This is a artistic representation. You can't actually see them all on the slide, but that's the basic idea. It's much denser and smaller than this. You have 400,000 or 800,000, and it's read off by the computer. We have different fluorescent levels corresponding to if there was the C or T, if it was binding or not. So how do we kind of visualize this when we have 400,000 or 800,000 methylation results? Well, we're used to looking at smaller things like 2 variables or 2 features. So I'm going to use a little analogy. So say we want to visualize some different musical artists. So here, actually I have 100 of them, but I'm showing the top snippet here. So we have 2 features of their music. We have a tempo, lyrical complexity. So I asked ChatGPT to make this up for me. It's probably, hopefully, somewhat accurate. It does, know the artists and understands numbers. But you may disagree with some of these things. So then, if we want to visualize it, it's pretty simple. We just make a XY plot, tempo on one, lyrical complexity on the other, and then fill them in. But then, of course, we're not seeing kind of the full complexity of different features. It doesn't really seem to give us clusters or anything. So, we might want more than 2 features. So now we have 10 features. So more specific tempo, lyrical complexity, harmonic complexity, energy level, harshness of the music, musicality of the vocals, is it rap or singing, era, if that's relevant, instrumentation, bass, things like that. And again, this is all generated by ChatGPT, so you can disagree if you want. And in fact, check it all. So when we have 10 dimensions, 10 features or dimensions are how we think of, is how we kind of describe these. We lose the ability to really visualize it. And I mean, we could go kind of 1 by 1, or 2 by 2 and kind of compare them, but then we can't see everything at once. This is, how you might do flow, where you're looking at all the markers on little 2D plots, and then you can gate and see which ones match up. But we kind of want a way to look at everything at once. So again, the concept, we think of each feature as a dimension, and the value is the position in that dimension. So the artist would basically be a vector of all these things. So here was Lil Wayne, here's Ed Sheeran's vector. And so we can think of each artist as occupying a point in 10-dimensional space. But again, the problem is we can't visualize that. So there are a couple of techniques for doing this. There's an older one, principal component analysis, which I'll leave off because I don't think that's used too much anymore. So T-SNE was a new one called that stands for T-distributed stochastic neighbor embedding. So there's a lot of information in the title there. But basically it moves the points so their distances in 2 dimensions reflect their distances in the higher dimension. So the axes don't really have a meaning, but the distances have a meaning and the relationships of the different points. So it's based on this, it has the word neighbor in here, so neighbor embedding means if they're neighbors in the higher dimension, in the 10-dimensional space, they're going to be neighbors in this 2-dimensional plot that we're making. The stochastic part of it means that it's not going to be exactly the same every time. If you run it different times, it might change a little bit because it's kind of randomly jittering the points a little bit. And it's an unsupervised learning algorithm, meaning you don't tell it what the answers are. You just give it the data, the vectors, and then let it sort everything out and figure out where they fit on the plot. So there's now a slightly better version of the T-SNE called UMAP, Uniform Manifold Approximation and Projection. So it moves the points again, so their distances in the 2D plot reflect their distances in the higher dimension. It just uses different math, so it's based on an edge graph, where the edge between points, where the edge strength reflects how close the points are, and then it replicates this in 2 dimensions. It's also stochastic, it's also unsupervised. It's better in most ways. It's faster, it preserves things better, it's more consistent if it's run multiple times, and you can rerun it on one additional case, whereas T-SNE, if you have new data points, you just have to rerun the whole thing. But when I wanted to show an example, it was easier to animate T-SNE, so I'm going to show you T-SNE here. So we start off with all of them initialized, which could be random or could have some basis to it, and then it's going to move them around again so that the distances of the different points reflect their distances in that 10-dimensional space. And then hopefully we end up getting more information. We get clusters of similar artists, similar genres, styles, time periods, all the different things it's taking into account. And again, one of the things to remember is you can't try to figure out, well, what is the x-axis and what is the y-axis? They don't really have any fundamental meaning. The only thing that's really preserved here is distances and hopefully relationships of, who's more similar, within a group and between different groups and things like that. So getting back to pathology a little bit, we kind of do something like this when we do different immunostains like CK7, CK20. You can think of different tumor types clustering in different regions. Again, we're only in 2 dimensions here. So 20 might be things like colorectal, 7 positive might be things like lung, adeno, breast, urothelial, squamous cell here. And so basically IHC is kind of a expression assay. It's a protein expression, which would then kind of be related to gene expression, which would then kind of be related to methylation. So you could think if you were testing methylation for these, you might be able to get a similar result. Of course, it's a little more complicated, but in theory at least. All right. So again, when we're visualizing these things, here's kind of a summary again. Each methylation value is a dimension, kind of like the artist descriptors were different dimensions. So sample is going to be all these different methylations. But you might notice if we're doing 800,000 methylation sites, that's actually more than the genes we have, which is like 20 or 30,000. That's because we actually have multiple methylation sites per gene. So it'd actually be more than just one for each. But in the end, it doesn't really matter for the algorithm. So you're going to have this large vector of 800,000 values for the different methylation data. And so each tumor is going to occupy a point in 450 dimensional space. And you're going to run your T-SNE, UMAP. This also is not real, still isn't actual brain tumor methylation data. This was just another animation I got online. At some point I'll get my own methylation data and can actually animate it. Again, sorts things out just to preserve those distances and data relationships. And so here's our methylation T-SNE plot of the different brain tumors based on the methylation data. I think this one was from the original paper if you want to look it up. But basically it sorts things out and you see pretty good clusters here, maybe better than the artist data, because it's more scientific, I guess, or there's more data. So you can see different things. Some of these abbreviations are a little hard to figure out, but you see different GBMs, the diffuse midline glioma, SFT, what else? ATRT, so is that astrocytoma, IDH mutant, maybe that's oligo. Just in case, I put in a list of abbreviations if you are wondering what everything is, so you can go back and forth if you're super curious. But anyway, it shows you the data relationships. It shows you it does a pretty good job of sorting things out and can maybe even give you different subclusters of different tumors. So again, this is an unsupervised algorithm. And it could uncover new information, such as the tumor has different subtypes. Maybe you have one tumor by histology, but then you find it has 4 different clusters. Maybe a different prognosis, or maybe it could be treated differently. You could find out that two tumors that you thought are different are the same tumor or a little more related than you originally thought. They usually just confirm what we already know, such as what's down here, the molecular subtypes of breast cancer, where kind of reflected things that were already known, like the ER/PR positive, the HER2 amplified, triple negative. So the T-SNE or UMAP are mostly used as visualization, and they can kind of give you an idea of where something fits. But when they were developing this, this is DKFZ, German Cancer Institute, Heidelberg, they wanted something more concrete to do the classification, and it's done by random forests. And I heard someone from their talk, and they were saying, you could use neural networks or something, and I think other places are using that. Maybe they will too. Random forests are just like a little more explainable, which is one of the reasons they picked that, and it's relatively simple to do, I think. Obviously things can get complicated as you try to tweak and perfect them. But we'll go into a little detail on how these random forests work. So forests are made up of trees, so the random forests we're talking about here are made up of decision trees. And this is a supervised learning algorithm where you actually do tell it what the answers are. You want it to specifically classify something and give you a confidence, and hopefully get the right answer, of course. So to explain decision trees, I asked ChatGPT to write a decision tree for what Taylor Swift breakup song you should listen to, and give me kind of a list here. I converted into a decision tree. So then you can kind of follow that through. So, okay, dumped by messy breakup, no. Anger and resentful, yes. So then you listen.
I'm telling you, we are never, ever, ever getting.
Okay, I hope they don't take this down. But then you can follow different paths.
Now we got bad blood. You know it used to be mad love. So take a look what you.
Okay, or maybe follow the path down here, but after
The
that.
players gonna play, and the haters gonna hate, baby.
Alright, you can't listen. I'd like to play the whole songs, but then they'll definitely take this video down. Alright, so again, getting back to pathology a little bit more, you could think about doing something like this for heme tumors. So, maybe you start by saying, of course you might get a large panel at once, but you might say, okay, is it 20 positive? Yes. Is it 5 positive? Yes. Cyclin D1, maybe it's mantle cell, something like that. And then again, like I said before, the IHC or the flow, you can kind of think of that as being related likely to methylation. So you could think of this as a methylation decision tree as well. If you were to do that, you might be able to do something similar. And then eventually you follow it down and get to different answers. So again, that's the decision tree idea. You're following it by different classifiers, and eventually you get to an answer. And this was my example for neuromethylation. Again, you don't necessarily know or care where they are. The decision tree is just kind of built by what works best, but you're looking at methylation of different points. Is it a certain number? Yes, no, follow different paths, and eventually you get to a diagnosis. And again, I saw a snippet of the real one, and it does actually, I made this one up of course, but it does actually look something like this, where you just, again, follow the different methylation values and eventually get to a diagnosis. So I won't go into this, but they're algorithms for making the decision tree itself. And given a data set, you can basically create the optimal decision tree to classify the cases that you have. But there's a catch. If you just create one decision tree, it's actually going to overfit the data and probably not be optimal for new cases. So you kind of want to tweak it, and what's done is to, instead of just having one decision tree, you have the forest of the decision trees. So you have multiple decision trees created with different components excluded. So maybe for this one we exclude 1,000 of the methylation sites or something like that, and then for the other ones we kind of exclude different things. And then we have a bunch of things that are a lot more robust, don't depend too much on any one thing. And then the trees vote on the consensus, so they'll say kind of what they think is best, and that can also give you a confidence. I think there's a number in the paper, there's a number I've heard, I forget what it is. I think it's like 1,000 decision trees making up the full random forest, but I'm not positive, so look it up if you're curious. So the DKFZ1 is available online, and you can submit things to classify. There may be some changes going on. I think they were trying to kind of make deals and possibly monetize it, but I know there's some changes that I haven't checked up in a little while. But again, however you access it, you can upload Illumina array data and then directly get the methylation results. So it'll run it through their classifier and give you an answer. If you want to run it clinically, of course, you would have to do your own validation, but you could still run it through them. They also have a soft tissue classifier and some other ones that they've been working on. So here's just an example of what the output might look like. So you get some data on it, classification, you would get a confidence too. So this one is from their paper where it looked at how things got reclassified. So, you know, if we're doing this, going to all this trouble, we're hoping to get some new information, maybe correct some things that were misclassified, or get some additional information like a subclass, something like this. This is showing some of the things that got reclassified, like anaplastic astrocytoma becoming GBM, what else? PA becoming diffuse midline glioma. So you can kind of see what happened to the ones that changed, and I think they tried to confirm anything that was changed to make sure it was correct or reasonable. Here's another example. This was just to show, this was a case of epithelioid GBM that got classified as anaplastic PXA. So that's one of the things I mentioned earlier, that it can kind of show you that maybe something that has a certain subtype is actually really part of something else. So, for instance, there's a paper showing epithelioid GBM's cluster with PXA, specifically anaplastic PXA, IDH wild-type GBM, which would just be, the normal one, and then a pediatric high-grade glioma. So, and it works well in a lot of cases, but there are limitations. It might be unclassified, insufficient, somewhere in like 5 to 10%. It might misclassify a tumor it wasn't trained on. If you give it something that's not in the training set, you don't know what it's going to do. It could call it something that's in there that just happens to be closest when it's following the paths and things. It can also, I hear, can do kind of random things sometimes, and you always have to correlate it with histology and other molecular IHC and all of that. And again, it wouldn't classify a novel tumor. Hopefully it would say unclassified, but it could potentially put it as something else. So the classic way this is done is the Illumina microarrays, the SNP microarrays. But there's another emerging tech, and I showed this briefly in the NGS lecture, but the nanopore is a newer tech. Well, been around a little while now, but it's slowly improving. It pulls the DNA strands through this membrane, through this nanopore, and by the current changes across it, it determines what nucleotide it was that passed through. But what it also does is it can give you methylation data. So the different methylation of the Cs can actually give you slightly different current change or voltage change. So you can figure out if they're methylated or not. So you can do this without a lot of the kind of classic procedure of the bisulfite conversion and all that. And the nanopore is very rapid as well. So this is one of the coolest things I've seen in molecular in recent years, is they could actually do it so quickly it can be done intraoperatively while they're doing a brain surgery to figure out what it is. I'd have to double check to see how long exactly it is, but it's kind of cool because it can actually, as it's running, it'll start to process the data and then slowly become more confident of the diagnosis call that it's making. Of course, I don't think it's standard of care. We'll see if it gets there. I don't know if we necessarily want to put ourselves in having to do these molecular frozen sections, basically, but it is still pretty cool, and we'll see where it goes. All right. That's all. Hope everything adds up, and if you have any questions, you could always try to reach out and find me. Pretty easy to find by the YouTube message or just looking up my name and finding an email.